Welcome to the Deep Dive, where today we'll be exploring the fascinating world of glass sponges.
Ooh, fascinating.
Yeah, these aren't the sponges you do the dishes with, right? We're going deep into the ocean depths of these.
No, not at all.
These creatures are basically living ornaments with skeletons made entirely of glass.
You got it.
And what's even more incredible is their strange biology and potentially ancient history.
Absolutely.
So today, we'll be taking a deep dive into glass sponges for all you intellectually curious amateur scientists out there.
Excellent.
We're going to uncover what research has revealed about these fascinating creatures, from their unique glass skeletons to their bizarre cellular makeup.
Sounds good.
So let's get started. What are glass sponges exactly?
Well, scientifically, they're known as hexactinellida.
Hexactinellida.
Yeah, and they're a class of sponges defined by these amazing skeletons made of silica.
Silica.
Yeah, think glass.
Okay.
These skeletons are built from these tiny interconnected structures called spicules, which are usually six-rayed, hence the name hexactinellida, hexa meaning six.
Makes sense.
And sometimes they can be four-rayed, but the six-rayed ones are the most common.
So it's like a microscopic scaffolding made of glass.
Exactly.
Very cool.
Right.
Now, I understand there's a bit of debate about where they fit into the whole classification of sponges.
There is.
Right. Are they with the rest or are they special?
Yeah, usually they're classified within the phylum Porphyra along with other sponges,
Classic sponges, yeah.
But some scientists, because of their unique characteristics, especially their unusual cellular structure.
But we'll get to that.
Yeah, we'll get to that. They've proposed putting them in their own phylum called Symplasma.
Symplasma.
Symplasma, yeah.
Okay, interesting. So if we were on a deep sea dive and encountered a glass sponge, what would we actually see? What do they look like?
Well, they often have this elegant cup-like shape.
Cup-like?
Yeah, kind of like a vase.
Like a vase?
Usually they range in size from about 10 to 30 centimeters in height.
10 to 30 centimeters.
Yeah, but some can be much larger, of course.
So it's not like a tiny little thing.
No, not at all. And the glass skeletons give them this really sturdy, lattice-like appearance.
But even though it's glass, it's actually quite strong.
It is surprisingly strong. A good example of this is the Bolosoma stalked glass sponge.
Bolosoma?
It looks really beautiful, like a delicate but strong glass vase.
Okay, and color-wise? Are we talking vibrant colors here?
Not really. They tend to be on the paler side.
Paler.
Whites, pale yellows, light oranges,
So blending in with the environment a little bit.
Yeah, I suppose you could say that.
All right, so we've got these glass skeletons made from these spicules.
Right.
How do they actually form this glass? It's like they're pulling glass out of the seawater.
Well, in a way, that's exactly what they're doing. They extract silicic acid from the seawater.
Silicic acid?
Yeah, which is a dissolved form of silica, and then they use that to build their spicules.
Okay, so is there like a special trick they use? How do they do that?
Well, some species, particularly those in the Euplectella genus.
Euplectella.
Yeah, also known as the Venus's flower baskets.
Oh, I've heard of those. They're quite beautiful.
They are stunning. They use a special protein called glassin.
Glassin, okay.
And this glassin acts like a catalyst.
The catalyst.
Yeah, speeding up the conversion of that silicic acid into the silica structures of their spicules.
So it's like their secret ingredient for making glass.
Exactly.
Oh,
It's
wow.
really fascinating how they've evolved this process.
Yeah, very cool. So it's not just about accumulating minerals. They actually have an active biological process going on.
Absolutely. It's very active.
Okay, so they're building these intricate structures. And in terms of their overall body plan, is it similar to a typical sponge?
Yeah, they have that fundamental sponge body plan, relatively symmetrical structure.
Symmetrical.
Yeah, and they have a large central cavity.
Central cavity, okay.
But in glass sponges, this cavity often opens to the environment through a sieve-like structure formed by those fused spicules.
So like a big open chamber in the middle.
Yes, precisely.
Now, let's talk about where they live. Where do we find these glass sponges in the vastness of the ocean?
They're mostly deep-sea dwellers, They prefer depths between 450 and 900 meters below the surface.
Wow, that's deep.
It is, yeah. It's well into the twilight zone, where sunlight barely reaches.
So are they like completely in the dark then?
Pretty much, yeah. But there are always exceptions, right?
Oh, of course, nature loves to break its own rules.
Yeah, we have Upsacus minuta, for example.
Upsacus minuta.
Yeah, it's a glass sponge that actually lives in shallower waters.
So it likes the sunshine a bit more.
Well, a bit more, relatively speaking. And then there are others that have been found even deeper.
Deeper than 900 meters.
Yeah, down in the abyssal plains.
That's incredible.
Yeah.
So are they found all over the world then?
They are. They've been found in all the world's oceans. They have a global distribution.
Global distribution, okay.
But interestingly, they seem to particularly thrive in the colder waters of the Antarctic and the northern Pacific.
So they like it chilly.
They do, they do.
All right, so we've got these deep-dwelling, glass-structured creatures. Now let's get into what really sets them apart. Their biology, particularly this thing called syncytia. What exactly is this?
Oh, that's one of the most fascinating things about glass sponges.
Yeah,
It's really a key reason why some scientists want to put them in their own phylum.
so it's a big deal.
It is. It is. You see, unlike most multicellular organisms, including other sponges,
Including other sponges.
yeah, much of a glass sponge's body is composed of this syncytial tissue.
Syncytial teeth.
Imagine this a single enormous cell with countless nuclei.
A single cell.
A single cell, yes,
Wow.
where the cytoplasm isn't divided by individual cell membranes.
That's like one giant interconnected cell.
Exactly. And this vast network is all supported by that framework of glass spicules.
That's incredible. So it's like a single, continuous, living entity at a scale we don't often see.
That's a great way to put it.
So how does this affect how they function? Does it change things?
Oh, absolutely. It has all sorts of implications.
Like what?
Well, even the cells that aren't part of this main syncytium, they still maintain connection.
Connection.
Yeah, they link to the syncytial body through these cytoplasmic rivers.
Cytoplasmic rivers, what are those?
Think of them like internal highways.
Highways.
Yeah, that transport nuclei, organelles, all the cell's internal machinery and other essential molecules throughout the entire organism.
So everything's connected.
Everything's connected. It's a remarkably integrated system.
So it's a radical departure from our traditional understanding of cells.
You could say that.
So how does this unique structure affect something as basic as how they eat?
Their feeding mechanism is also very distinctive.
Okay, how so?
Well, they don't have the typical choanocytes that most sponges use.
Choanocytes.
Yeah, those flagellated collar cells that create the water currents.
Oh, right.
Instead, glass sponges have these things called choanosyncytia.
Choanosyncytia.
Yeah, it's a mouthful, I know. These are further syncytial regions organized into these bell-shaped chambers.
Bell-shaped chamber.
And lining these chambers are these collar bodies,
Collar bodies. Okay.
which are basically like choanocytes with a collar and a flagellum.
Right.
But they're not individual cells. They're part of that multinucleated tissue.
So they're all working together.
Exactly. And the beating of these flagellas all across this tissue draws water through the sponge's body, allowing them to filter out food.
Like a coordinated feeding effort on a massive scale.
Exactly.
Very cool. Now, can they do other sponge-like things? Like, can they contract their bodies?
Actually, unlike a lot of other sponges, glass sponges can't contract.
They can't contract.
Nope. This is probably because of their rigid skeleton, the glass.
Right, that makes sense.
And their syncytial organization, which isn't really built for muscular contraction.
So they're more like stationary filter feeders.
Pretty much, yeah.
And I read that they can actually sense and respond to their environment really quickly, almost like a nervous system, which is not something you'd expect from a sponge.
You're right. That's another amazing adaptation.
What's going on there?
They have this unique system for conducting electrical impulses across their syncytial body.
Electrical impulses, wow.
Yeah, and this allows them to respond quickly to things happening around them.
So they're more aware than we might think.
Much more aware.
Do you have an example?
Yeah, there's this species called Rhabdocalyptus dawsoni.
Rhabdocalyptus dawsoni.
When it detects sediment in the water, stuff that could clog its filtering system,
Yeah.
it stops feeding immediately.
Wow, so it's like a built-in defense mechanism.
Exactly, and it's all thanks to these electrical signals. It's amazing.
It really is. Now, you mentioned Euplectella earlier, the Venus's flower basket.
Yeah, beautiful creatures.
They are. Aside from their amazing shape, do they have any other interesting biological features?
They do. At the base of their skeleton, they have this tuft of long, hair-like fibers.
They're like fibers.
They can be quite long, from 50 to 175 millimeters.
Wow, that's pretty long.
And they're incredibly thin, like human hair.
And what are those for?
We're not entirely sure, but we think they help anchor the sponge to the soft sediment on the seafloor.
So like roots, holding them in place.
Exactly.
Fascinating how these delicate structures can be so strong and functional.
It is, isn't it? And some glass sponges even have symbiotic relationships with other organisms.
Like little partnerships.
Yeah, exactly. There's Vazella portulaceae, for example.
Vazella portulaceae.
It hosts all sorts of microbes in its tissues.
Microbes.
And these microbes help with things like nitrification and denitrification, which are key parts of the nitrogen cycle.
So they're helping the sponge survive.
They are, especially in those low oxygen environments in the deep sea.
It's amazing how interconnected everything is down there.
It really is.
Now, one of the things that I find most intriguing about glass sponges is their potential for extreme longevity.
Oh, yes, that's mind-boggling.
Are they really the longest-lived animals on Earth?
Well, they're definitely contenders for that title.
So how long are we talking?
There was a study that modeled the lifespan of Scullymaster jubini.
Scullymaster jubini.
Yeah, and they initially estimated an age of up to 23,000 years for a single individual.
23,000 years.
It's incredible, right? But later research, taking into account sea level changes,
Sea level changes.
yeah, revised that estimate down to around 15,000 years.
Yeah. Okay, so still an incredibly long time.
Still an incredibly long time. To put that in perspective, that individual could have been alive during the last ice age.
That's amazing.
Ugh.
So they've witnessed so much history.
They really have.
Now, you mentioned they're mainly deep-sea creatures, but
Yeah.
there are those rare exceptions in shallower waters.
Right.
What's the story there?
It is true that shallow water occurrences of glass sponges are rare globally,
Globally, yeah.
but there are some interesting exceptions in Antarctica, for example.
Antarctica.
There are 2 species that live as shallow as 33 meters below the ice.
That's pretty shallow.
Yeah, much shallower than usual.
Yeah.
And in the Mediterranean Sea, there's a species that lives at about 18 meters deep.
18 meters.
But it's in a cave environment where deep, cold water wells up.
So it's like a little pocket of deep-sea conditions.
Exactly. So it seems it's not just depth, but also the right combination of temperature, water chemistry, and stability that they need.
They've got their preferences.
They do.
Now, something else I learned that really surprised me. Some glass sponges can actually form reefs.
Oh, yes, the reefs.
It's not something I usually associate with sponges, you know.
Yeah.
I think of coral reefs.
Yeah, coral reefs are more common, but these glass sponge reefs are a remarkable discovery.
So how do they form these reefs?
Well, certain species of glass sponges can fuse together over time.
They fuse together.
Yeah, creating these large three-dimensional structures, the reefs.
And where have these reefs been found?
They've been found off the coasts of British Columbia, Southeast Alaska, and Washington State.
So in the Pacific.
Yeah, in the Pacific. And there's a lot of research going on to study them, like the Sponge Reef Project.
The Sponge Reef Project.
Yeah, they're doing some amazing work.
And these aren't just small patches, right?
Oh, no, some of them are quite extensive.
Yeah. How extensive are we talking?
The reefs in Hecate Strait in British Columbia, for example, can be up to 7 kilometers long
11 kilometers.
and 20 meters high. It's like an underwater city.
That's massive. What's even more remarkable is that we used to think sponge reefs went extinct millions of years ago.
Yeah, back in the Jurassic period.
Jurassic period.
So finding these living reefs today is like discovering a lost world.
It really changes our understanding of how reefs form and evolve.
Absolutely. And, some of these reefs even mimic the appearance of coral reefs.
How so?
Well, the species Cerastasia oculata.
Cerastasia oculata.
Yeah, it always has these symbiotic zoanthids living on it.
Zoanthids, what are those?
They're these small anemone-like animals.
Okay.
And because of this partnership, the glass sponge reef ends up looking a lot like a coral reef.
So it's like an ecological mimicry.
Exactly. It's a great example of convergent evolution.
Very cool. And I also came across mentions of glass sponges on shipwrecks and possibly a wider distribution in the Pacific than we thought.
Yeah, those are some interesting recent findings.
Tell me more.
Well, they've found glass sponges growing on the wrecks of HMCS Saskatchewan and HMCS Cape Breton.
Those are Canadian ships, right?
Yeah, they sank off Vancouver Island. And this shows that glass sponges can colonize new hard surfaces in the deep sea.
So they're adaptable.
They are. And then there's the discovery of these zoanthran species.
Those anthers species.
Yeah, they're specifically associated with hexactinellids, and they've been found near Japan and possibly Australia, which suggests that glass sponges and their communities might be more widespread in the Pacific than we realized.
So there's still so much to discover.
Oh, absolutely.
So given their delicate structures in deep-sea habitats, are glass sponges facing any threats?
Well, for most deep-sea glass sponges, their remoteness offers some protection from human activities.
They're out of reach.
Yeah, but the glass sponge reefs off British Columbia are a bit different. They're more vulnerable.
Why is that?
Well, they can be damaged by bottom contact fishing gear,
Bottom contact fishing gear.
like trawls and dredges, which can destroy their fragile structures
So they're more easily disturbed.
much more easily.
But I understand there have been some efforts to protect them.
Yes, thankfully, the Canadian government has established a marine protected area in Hecate Strait and Queen Charlotte Sound.
So that's good news.
It is. It
Oh.
is. And there are regulations prohibiting bottom contact fishing near the reefs.
That's great to hear. Now, what about climate change? Is that affecting them?
That's a crucial question, and research is still ongoing.
Okay.
Experiments on Africolestes vastus, for instance,
Africolestes vaste.
yeah, have shown that warmer water and ocean acidification, which are both results of climate change,
Right, right.
can weaken their skeletons.
So they become more fragile.
Yeah, more susceptible to damage. But there have also been some surprising findings.
Oh.
Yeah, after the Larsen A ice shelf collapsed in Antarctica in 1995.
That was a big event.
It was, and it was linked to climate change. But after the collapse, they actually saw an increase in the size of hexactinellid reefs in the area.
So they benefited from it.
In a way, yes. It seems their response to climate change is complex, and we're still trying to understand it.
It always is with nature.
It always is.
Now let's talk about their evolutionary history. You mentioned they have a very ancient lineage.
They do. They do. The earliest confirmed glass sponge fossils are from the early Cambrian period.
Early Cambrian, that's over 500 million years ago.
That's right, over 500 million years ago. And there's even a possible relative called Helicolicellus.
Helicolicellus.
Yeah, from the late Neoproterozoic era, which is even older.
Wow.
So their lineage is incredibly ancient. And actually, their fossil record is pretty rich compared to other sponges.
Why is that?
Well, their glass spicules are much tougher than the softer sponge and fibers found in other sponges.
They last longer.
Exactly. They fossilize much better. So we have a good record of their history.
That's fascinating. So they've seen a lot of changes over the eons.
A lot of changes.
And despite their unique cellular structure, are they still filter feeders like other sponges?
They are, at their core, they're still filter feeders.
So that basic mechanism has endured.
It has. It's a very efficient way to feed. And it's been around for a very long time.
Very cool. And finally, how are glass sponges classified within the whole sponge family tree?
Well, within the phylum Porifera, there is a separate class called hexactinellida.
Hexactinellida.
And this class is divided into two subclasses, Amphidiscophora and Hexasterophora.
Amphidiscophora and Hexasterophora.
It's a mouthful, I know,
It is.
but it's based on the shape of their spicules.
Okay.
And then these subclasses are divided into several orders,
Several orders, all right.
including Amphidiscosida, the extinct Hemidiscosa, and Reticulosa.
Extinct. Wow.
Yeah, long gone. And then there's Lichniscosida, Lysocinacida, Saprolophora.
Okay, keeping track here.
And lastly, there's the family Dactylocolicidae,
Dactylocolicidae.
which is currently classified as Incertisades,
Incertisades.
which means we're not quite sure where it fits in yet.
Still a mystery.
Yeah, science is always a work in progress.
So, a long and complex history for these creatures.
It is. It is.
Well, this has been an absolutely incredible deep dive into the world of glass sponges,
I agree.
from those delicate glass skeletons to the incredible depths they live in.
And their unusual biology.
Absolutely. And their potential for extreme longevity.
Thousands of years.
It's mind-boggling. And then the fact that they can build reefs.
Like underwater cities.
It's amazing. And their ancient origins stretching back hundreds of millions of years.
They're truly remarkable creatures.
They really are. And they show us that even in the most extreme environments, life finds a way to thrive.
And thrive in extraordinary ways.
And on that note, I want to leave our listeners with a final thought.
Okay.
Considering everything we've learned about glass sponges, their syncytial nature, their incredible lifespans, what other secrets might they hold?
What other secrets?
Yeah, what could they teach us about evolution, about multicellularity, about aging, about cellular repair?
It's fascinating to think about.
It really is. So much to learn from these ancient creatures.
I agree.
Thank you for joining us on this deep dive into the fascinating world of glass sponges.
It's been a pleasure.
We'll see you next time on The Deep Dive.
See you then. Bye.